Hi, everyone. I'm Beth Orcutt, in case you haven't met me before. I am a senior research scientist at the Bigelow Laboratory for Ocean Science in Maine. I'm a deep sea microbiologist by training, as is the co-speaker today, Julie Huber from Woods Hole Oceanographic. We are the co-directors of the COBRA project that Julie talked about at the beginning. Oh, Rosalind, just want to make sure we're recording. Are we recording?
Yes, we are.
Great, thanks. So we're going to record today's presentation. We will turn it off for the Q&A so that you feel unencumbered to ask your questions. And really what we want to focus on is trying to define what are some of the important microbial ecosystem services that are happening in the deep sea. What might be the best indicators for those services? What do we know about the baselines of those services? And do we know anything about thresholds for harm of those services? But before I get started with that topic, I want to make sure we're all on the same page about microbes, because you may not think a lot about microbes in the ocean. And so I just want to make sure we're talking about the same thing. And you might have a reaction that microbes are bad, but I'm going to remind you that microbes do lots of really important things to benefit humanity. Here are some delicious foods that are made by microbial processes acting on natural materials through fermentation and processes like that. So microbes can do really beneficial things for us, and they're in all environments, and many of them are good, some of them are bad. And really, microbe is an operational term. It means you need a microscope to see it, but it encompasses a very wide diversity of life. We often think of microbes as synonymous with bacteria, but actually there's whole other domains of life, like archaea in there. We also have viral particles that are part of the microbial realm, microbial protists, fungi, yeast. All of these things are kind of encompassed in the term microbe. So that's an important thing to remember, that it's a very diverse group of organisms that are microscopic. And as my little analogy about food reference, right, microbes provide services to us, and they also provide services on the seafloor. And we really want to understand how those services might be impacted by deep sea mining. So again, if you don't know much about deep sea mining, just a little introduction. This is the idea that there are mineral resources on the seafloor that might have high metal content to make them a valuable economic resource. And so you would use different technologies to try to lift, collect those metals and minerals and bring them up to a surface ship. That might be done on abyssal plains where you have nodules on the seafloor. It might be done on seamounts. It might be done at mid-ocean ridges where you have hydrothermal sulfides. And this collection activity might create disturbance both on the seafloor as well as in the water column, where sediment is remobilized. Maybe the minerals themselves are broken down into small pieces. And so that might have impacts around the deep sea area. As Julie mentioned at the start, right, the COBRA project really thinks about all those different environments, whether they be the active sulfides, seamount ecosystems, or nodules in the abyssal plains, and how human activity in those environments may influence those ecosystems, but also trying to address the knowledge gaps about life in those systems, and particularly microbial life, since that's something that we are experts in. This figure I'm showing you came from an information sheet that the Deep Ocean Stewardship Initiative put together, defining some of the services that come out of the ocean and the deep sea. And the ones that are highlighted in red are services that microbes play a really important role in, whether that's genetic diversity, their role in chemical cycling. But this actually doesn't paint the full picture of all the things that microbes do. So a goal that we had was to try to give more definition to what microbes do and provide as services in the deep sea ecosystem. So I'm going to now turn it over to Julie to explain what some of these services are.
Great. So yeah, let's dive into the ecosystem services that are provided by microbes in the deep ocean. And remember, these ecosystem services are really defined as contributions to human welfare from the environment or ecosystem. And so through our work over the last year or so, we've identified 10 key microbial ecosystem services in the deep sea. 5 of them are regulating services, 4 supporting, and finally 1 for provisioning. And I'm going to walk through some of these services just to help you understand them. And then we'll talk about why they're important to understand in the context of deep sea mining, and then turn to our most recent work in determining the values for thresholds for harm to some of the services. So the first service is synthesis of new organic carbon. So on land, we're all familiar with this process of primary production using photosynthesis. Plants, right? They use energy from the sun to make carbon. But in the deep ocean, carbon can also be fixed in a similar way, but instead of using light, they use chemical energy. So this is also referred to as chemosynthesis or dark carbon fixation. And on the seafloor, in a lot of different environments, microbes perform chemosynthesis, and they use a lot of different chemicals for this energy source. And so this means that new carbon is being produced, and it's entering the biosphere and supporting the entire food web. Beyond that, feeding into the food web, we also have the ecosystem service of basically the microbes themselves are food. There's a lot of different things that either graze on microbes or they filter them out of the water, and that provides this service of being food for the food web. An example of a regulating service microbes provide is through carbon capture in climate. And we're going to talk about this in more detail, but basically over long geological timescales, microbes rework the carbon pools of both organic and inorganic carbon in marine sediments. And that's really a key variable in thinking about how much carbon is actually getting captured in deep sea sediments versus how much is getting respired back into carbon dioxide and released back to the environment. So microbes are really controlling that balance, and that provides a key carbon capture service in removing carbon for the atmosphere. And depending on where you are in the ocean, like a carbon poor sediment like we have in nodule-bearing environments versus a coastal environment, that balance will be different. So in coastal environments where you have a lot of organic carbon, microbes might not be able to fully respire the carbon, so more of it gets stored versus out in these abyssal plains. And we'll talk more about how this scales in just a minute. And finally, the deep sea is just this immense reservoir of genetic diversity. Almost every habitat we look at hosts these really unique microbial populations. And as humanity increasingly looks to genetic novelty for the development of new drugs, new natural products, the deep sea has really become a provider of genetic diversity that can be exploited. From an ecological point of view, genetic diversity is also just a really key factor for an ecosystem's ability to respond to change. And we'll talk more again about what we know about that later. So we think these services are really important. And as we'll show you, though, we're really lacking data on a lot of their value, both in terms of the service they provide, as well as their economic value. So we can't really assign a dollar value to a service without knowing the magnitude of its baseline. So this is really important to consider when we're potentially removing value from the ocean via resource extraction. So I'm going to hand it back to Beth to talk about why this is important.
Great. So beyond us having a passion for microbes and wanting to understand these questions, this work is also motivated by some urgency in that the organization that is tasked to manage mineral resources on the seafloor underneath international waters, the International Seabed Authority, is currently trying to draft guidelines for the procedures for potential deep sea mining in those areas. And part of that work is to develop binding values for thresholds to harm to services and diversity and things like that. And so that effort is being led by the Legal and Technical Commission of the International Seabed Authority. And part of that work is done by an expert group. Disclaimer, I am serving on that group. I will not talk about what that group is doing, but more about how work as scientists can feed into that work. So there's a definite need for this at the international level to inform the development of these regulations. And within COBRA, we recognize that there was a big gap in knowledge and translation of that information about ecosystem services, particularly that are provided by microbes, to inform the kind of work of these groups. And so last year, we brought together a group of subject matter experts, shown in this picture here, to try to tackle this topic and say, what are those services? What can we say about their baselines, about their best indicators? If we know anything about thresholds to harm, to try to address that science to policy gap. And this work was funded by COBRA to bring this group together, and it was done in collaboration with an event hosted by the Deep Ocean Observing Strategy to bring experts together to talk about issues related to the deep ocean and how observing can be accelerated to address those knowledge gaps. So what our group did is kind of think about the deep sea and these nodule-bearing abyssal plains. And when I look at a picture like this, I think of all the invisible microbial life that is on the nodules, that is living in the sediment, that is in the water, that is supporting these animals. And what we wanted to do in this workshop is really come away with, can we start developing baselines for some of these services? So I'm going to hand it over back to Julie to talk about what are we learning about the magnitude of some of these services.
Yeah, and I should mention the 10 services that we have here. It was in that room on that whiteboard that we decided these are the 10 services. And now we're going to go through some examples of what we've done so far with respect to finding value ranges for these services, how to measure them, the thresholds. And we're going to start with this service I mentioned, synthesizing new carbon via dark carbon fixation. So you're going to be seeing this table over and over again. So I want to walk you through it. To the left is the ecosystem service we're addressing. And then the second column is based on our search of the published scientific literature, the baseline range for that service in sediments, ideally in nodule fields when such data is available. The next column, the best indicators, is really the method for measuring the surface. And then the next column shows whether or not that indicator is in current ISA guidelines for the environmental impact statements for contractors. Of course, keeping in mind the work that the intersessional expert group that Beth is part of that still needs to be done. And then that final column shows whether or not there is a known threshold for when this service is harmed. So now that we've explained that, let's talk about this particular service, primary production. So we have some data. We have some baseline measurements in sediments from nodule field shown here. And one really important point about measuring primary production in the deep sea is it's a pain. It's a very intense method. You have to use either stable or radioactive isotopes. And it's a complex process. It's pretty slow, and you really have to track not only the tracers, but also need to know the concentrations of both that organic and inorganic carbon pool. And that makes it really labor intensive and computationally intensive. The current indicators in the guidelines include those carbon compound pools, but they do not include any isotopes for rates, and nodules are not included in that measurement. So for setting a threshold, we do have some data to look at. This is from some really elegant work by our German colleagues in the Peru Basin nodule field, where a disturbance experiment was carried out in 1989. They basically removed nodules similar to mining technology being spoken about at the time. And then they've been following habitat recovery. And so 26 years later, they went back and carried out assessments of a lot of different factors. And what's shown in this slide is dark carbon fixation, so that primary production by chemosynthetic microbes. And all the way there to the right, circled in red, is the carbon fixation rate in the most impacted part of the sediments. And that's compared to that reference over there in green. And so they found that microbial dark carbon fixation was reduced about 95% compared to the reference. And they estimate it would take about 50 years to return to undisturbed levels. So in this example, you know, it's clear this service was harmed through the process of nodule removal. So let's look at another example of microbes serving as food for the food web. Again, we can return to our table, and here we have actually a lot of data. People count cells in sediments a lot. That's the most basic metric for answering how much food is there, is counting how many microbes you can see using a microscope. A more sophisticated method would be trying to trace the uptake of those microbes into benthic animals, but that again requires incubations, and it's not at all a routine measurement. Cell counts are included in some of the guidelines, but only for sediments and seawater, again, not for the nodules themselves. And it only includes prokaryotic life, bacteria and archaea, not any of the other members of the microbial food web. And again, for setting a threshold, we can turn to that German disturbance study. So this is the exact same work. The two figures on the left are identical, but now the figure on the right is showing microbial cell abundance on that y-axis. And again, you can see that microbial cells 26 years later were still reduced by about 50% from that fresh track. So here, we're toying around with the idea of setting a threshold of 50% means harm has happened. So maybe 25% reduction in biomass means something as bad is starting to happen. So here's where having studies like these are really important for setting these thresholds. So I'm going to hand it back to Beth for this next part.
Thanks. So how do we consider the role of the service of carbon capture that's happening in the seafloor? So as Julie talked about at the beginning, right? Organic carbon that rains down through the water column or is created on the seafloor is in the sediments, and microbes basically regulate how much of that will be respired back to carbon dioxide versus how much will actually get buried in the seafloor. And in the abyssal plains, there's not a lot of carbon in those sediments. And so on a kind of per teaspoon basis, there is not as much carbon capture potential in abyssal plain nodule sediments as there is, for example, compared to a teaspoon of coastal organic rich sediment, where microbes basically are overwhelmed by how much carbon there is. They run out of energy to breathe that carbon, and so that carbon escapes respiration and is buried. But that also happens in nodule fields, just at a smaller scale locally. But when we consider the amount of spatial area that nodule mining might impact, this is where it might become a little tricky. So if you were to say, how much carbon is in the upper 10 centimeters of sediment in the amount of area that might be mined by a contractor, and that's estimated to be between 200 and 300 kilometers squared. It's kind of driven by operational efficiency versus also how much money the nodules might be worth. So we come up with an estimate that roughly 41,000 tons of carbon are buried in that upper 10 centimeter layer in a 300 kilometer square box. So that's how much carbon naturally kind of exists on the seafloor, and that has the potential to be buried for carbon capture. Now, if mining were to occur, that will create a disturbance. That upper 10 centimeters of sediment might be remobilized into the ocean. Much of it will resettle on the seafloor. Some may stay up in the water. And where this process becomes important is that microbes may react to this remobilized organic carbon, and they might respire it either in the water or in the sediment. Understanding if that service is changing over time requires things like measuring the total carbon concentration, but also doing Lander measurements of oxygen gradients in sediment so that you can see if the gradient of oxygen is changing compared to a reference. And is that an indication that more of that carbon is being respired or it's staying the same? So both of those methods are actually in the indicator guidelines, but the Lander measurements in particular are not trivial. So let's continue this thought experiment for a little bit. So I explained to you where we come up with this 41,000 tons of organic carbon that could be remobilized per contract area per year. And remember that that represents actually hundreds to thousands of years of sediment burial and carbon capture that would be remobilized within a year time frame. Now, if all of that organic carbon was respired and converted into carbon dioxide, that would be the equivalent of 150,000 tons of carbon dioxide that has the potential to be remobilized per contract area per year. And if these are numbers that you don't often think about for scale, this is roughly equivalent to the CO2 emissions from small island developing states like the Cook Islands or Tonga in 2022. Now, I'm not saying that mining will create that much carbon dioxide. I'm saying it has the potential to create that much carbon dioxide if microbes are able to respond to that refresh of organic carbon and respire it in a way that they would not have done if the sediment had not been remobilized. So that's really a key point here, is that we need to actually understand how much of that carbon is going to be remineralized. And it requires those methods that I was discussing to try to measure that. When we think about how much deep sea mining might happen on a larger scale, right, this could be an important thing to keep in mind. Again, it's nowhere near the biggest carbon emissions on Earth that we need to keep track of. It may not even be as much as the emissions that come from the ships themselves that are out there mining, depending on the technology that those ships are using. But it is something we think about with this emerging field of marine carbon dioxide removal, right, of what is the price of that carbon? So happy to talk more about this. So I'm going to come back over to Julie to talk about another service.
Right. So this process that Beth just mentioned, it not only respiration not only generates carbon dioxide, but in many cases, oxygen is consumed by microbes in that process. So now we want to turn our attention to this service of microbial oxygen cycling. So microbes can still respire without oxygen, but we're in this case thinking about what happens to animal life when you lose too much oxygen. So dissolved oxygen is essential for animal life in the ocean. They have to be able to respire, to grow, to move, to reproduce. And these decreases in oxygen can limit both animal abundance and their overall growth. So we have pretty good measurements of oxygen in sediments, and I'll show you in a second. There's a lot of variability depending on where you are. The best indicators for this service are these in situ sensors and lander-based incubation chambers that Beth mentioned, which makes it a challenge. It can be a challenging measurement. This service is included in the guidelines, but mostly for water, and only rarely have we seen it actually reported in environmental impact statements for sediments. However, based on a lot of animal studies that have been done in very different contexts, we do have a threshold for harm with respect to animals, and that's between about 8 and 40 micromolar oxygen. I did also want to mention that we need to think about oxygen concentrations because that's also an important control on the mobilization of metals. So decreasing oxygen concentrations can mobilize more metals, and that's another thing that we should think about considering disrupting the seafloor. So I just wanted to show you, in case you've never seen these oxygen profiles, this is 2 different profiles from the CCZ in the top 30 centimeters or so of the sediment. And a lot of the existing guidelines, and Beth mentioned this, they talk about measuring things down to about 10 centimeters. So we'll draw that line where you can have, and you can see right away, we have very different oxygen concentrations already at these 2 sites. And then we can add that threshold, that 40 micromolar line, which is kind of the high end of minimum of what animals need. And so hopefully what you're getting just without any disruption, the animal communities here, their ability to survive in this sediment is going to be quite different because of these oxygen profiles. And because of these different profiles, microbes in the sediments are likely going to respond to disturbance differently. So on the side on the left, the organic content is probably really low already. There probably isn't that much to eat. So a disturbance with respect to oxygen might not matter that much with respect to that drawdown. But at the site on the right, the organic content might still be high. And so if oxygen is reintroduced via disturbance, all of the oxygen locally could get consumed. Again, thereby impacting animal life. So the final example that we also wanted to talk to is the provisioning service of genetic diversity. So we're still working on setting our baseline range for this in sediment. We have a lot of data to draw on. I'll show you that in the next slide. But the best indicator of this service is using DNA sequencing technology, both to measure the number of microbial species, but also their functional diversity potential. And we refer to this as tag sequencing or 16S ribosomal RNA gene sequencing to get to the who and how many. And then we refer to the functional diversity potential as metagenomic sequencing. So as far as the indicator guidelines, taxonomy sequencing is currently included. It's only included for the sediments and seawater, not nodules, and there's no metagenomic sequencing included, and we don't yet have a threshold for this service. So I wanted to show you some examples, one that is published, one unpublished. to see kind of where things stand with respect to the biodiversity and the genetic diversity in these environments. So the figure on the left came out a couple of years ago, synthesizing the 144 data sets available at the time, looking at the 16S ribosomal RNA gene sequencing data. This is something called a non-metric multidimensional scale ordination. Basically, the further apart the points are, the more different they are. The shapes are showing bottom seawater, sediment, and nodule, and the colors are just from 3 different cruises. So as you can see, seawater is very different from the nodules and the sediments, and then there's a lot of overlap between the sediments and the nodules, which makes a lot of sense because these nodules are sitting right on top of the sediments. So this data, this older data, basically shows these are distinct communities in these different habitats. So within our COBRA team and led by Gus Ramirez at Cal State LA, we've been analyzing even larger data sets for the paper we're working on. And what I'm showing you here is something called a Venn diagram that is showing nodules, again, sediment and seawater, but this time the size of the circle is scaled to the number of species within each substrate type. And then that overlap between them is also scaled. So you can see there's a lot of novel biodiversity and genetic diversity within each habitat type, and removing anything from this ecosystem will remove biodiversity. And there is some overlap, but there are also these completely unique parts that we need to keep in mind. So back over to you, Beth.
Thanks. So this is revisiting that DOSI infographic, but superimposing on it so these 10 services that we have identified that will be described in our report. We've talked to you about a few of them: the support of organic carbon creation, the support of microbes as food, microbes supporting oxygen cycling. Microbes also support nutrient cycling. We didn't have time to talk about that today. On the regulation side, in addition to regulating carbon capture and sequestration potential, microbes also, we think, play a really important role in regulating toxic substances, whether those be metals or other components in the environment. They play an important role in the emission of greenhouse gases, and in nodule-bearing sediments, the greenhouse gas of most concern is nitrous oxide. Microbes play a really pivotal role in the regulation of that gas, and that might change with mining impacts. We also think that microbes play a really important role in chemical signaling for successful larval settlement. This is a relatively new field. We haven't talked, there's not a lot of much data on it, but it's something that we will talk about in our paper. And in addition, microbes also regulate animal health. This is well known in hydrothermal vet systems where you have really unique animal-microbe symbiosis. It is less well known in nodule settings, but it is likely that that plays an important role. And importantly, because we know so little about it, we also don't know when animals acquire their symbionts. What part of their life cycle are those symbionts acquired? And could that be impacted by mining? So again, revisiting those 10 services and thinking more broadly, do our current regulatory documents give the right methods that we would capture these services, that we would capture the right indicators? Are those indicators, if they are in the guidelines, are they commonly being reported in environmental impact assessments that are being submitted to the International Seabed Authority? Whether or not we have thresholds that are known for ecological effect-based harm, thresholds of harm, is it possible to define some management thresholds to look for those effect thresholds to see if harm is occurring? And so hopefully you see in this table right away that there's a lot of red, which is an indication of the knowledge gaps that we still have about these 10 services. And in particular, some of these services related to animal health and regulation of larval sediment, we have really big knowledge gaps about these services, what their baselines are, how we would appropriately capture them to see if those services were being harmed, et cetera. So all of these are going to be described in more detail in this report that is forthcoming. But hopefully this gives you some key takeaways about where we're at with this. So Julie, do you want to close this out?
So to close it out, some key takeaways that I hope you can walk away from is that microbes perform these really important 10 ecosystem services in the nodule-bearing abyssal plains. Most of these services are missed in baseline studies and impact assessments. We would love to work with economists and others to try to use these baseline values for economic valuation studies. And there are still these really critical knowledge gaps, as Beth just mentioned, especially with respect to genetic diversity, carbon burial, toxicity, animal health, and larval sediment. One big takeaway that we also wanted you all to walk away from that really quickly emerged in this room of experts is that many of us collect samples in the field and then we bring them back to our home labs to do these assessments. But we really need to push the envelope here in developing shipboard methods to assess some of these changes to microbial services so that we can do it in real time to indicate potential harm. Some of this will be relatively straightforward. In other cases, we really need to innovate and try to be more accurate so we can do this. You know, the CCZ, for example, is very far from land. And so we really want to push the envelope on this. And finally, just to remind you that this is really timely. So Beth just got back from an IEG meeting, and this is a really aggressive timeline to set these deliverables for the binding environmental threshold values. So if you think you have something to contribute, please reach out to us. And finally, I just want to thank this really exceptional group of people who sat with us. This was a view behind us, but we were in a room for 4 days working on this. Beth and I just have the privilege of being the messengers. They've been working really hard on everything that we've shown you today. And we really appreciate, we see a bunch of you online today. So thank you for being part of this. And with that, we're happy to take any questions.